Thanks for the invitation to present my research today. So I made the title a bit more specific. Now I'm talking about NTIMP2 and MP9 interaction, how we use deep learning trained on deep mutational scanning data to allow the prediction of the affinity landscape. This work was done by my student Ruth Moshe. She couldn't make it, so I'm presenting the work for her. And this was done in collaboration with my collaborator Niv Papo and his student Yaron, who did all the experiments. So introduction, I think we all know in this room what proteins are. It can be represented as a string of amino acids, or in secondary structure, or folded by solved structure, or predicted structure. And one subtype of proteins are called enzymes, and they interact with a substrate, and through this interaction they can, for example, break the substrate into products. So enzymes perform very important roles in the cell, and are also natural inhibitors that compete with the substrate for the binding site of the enzyme, and then they prohibit or prevent the binding of the enzymes. Now, these natural inhibitors can be used to engineer them and improve the prohibition of the substrate binding, and so can be used for therapeutic applications. So we are interested in engineering inhibitors to have improved affinity or lower affinity to the binding site. And protein-protein interaction, and specifically the type of interactions which I refer to, which are the enzyme inhibitor interactions, play essential many roles, and they have been studied for years in this audience and in academia in general. There's lots of experimental data, and based on this data, there are many prediction methods. Here I'm just citing one review paper. Of course, there was a breakthrough just a few months ago with AlphaFold 3 that can give us very accurate predictions of protein-protein interactions, or not that accurate. But now we're interested in a specific type of prediction. It's not about two proteins or just two proteins in a complex, but the effect of variants, so single amino acid mutations or double mutations on the interactions. And here I think that the methods are more struggling. And for this aim, researchers have developed experimental techniques, experimental assays that can collect informative data so we can train models to perform these predictions. So the experimental technique, and I'll guide you through it in this illustration, I will be using in my study. It's called deep mutational scanning. It's an established technique. Many labs do it, as well as the lab of my collaborator. We start with a library of variants you see here, and this library could be, for example, a combinatorial library where we make sure we have each single amino acid mutation over the positions, or it could be a random with error-prone PCR or any type of library. Then these protein variants, once they are expressed and purified or for use surface display or whatever technique, are put under some selection, a selection for a specific function that we want to measure. These protein variants and their abundance is measured both before the selection and after the selection. And this can be done, for example, using high-throughput sequencing, we can sequence the DNA coding templates. Then we look at the protein. Here, for example, you can see the red variant, how it increased in frequency before selection and after selection, meaning it had an improved affinity or function for the specific function that we selected for, as opposed to the purple, for example, purple variant, where we saw a reduction in the frequency. So this is a very effective experimental technique to provide high throughput measurements of the effect of variants on a specific function. And the very common and established label that people use, label for the machine learning algorithm or just as a score from the experiment, is called an enrichment ratio, where we look at the frequency of the variant in the sorted library, meaning after selection, over the frequency in the naive library, meaning the initial pool of variants. So this is the technology and the data that we are going to use, but there are a few limitations and gap of knowledge when we use DMS data. So most of the studies focused on variants that span a small range of affinities, and we are wondering if we can predict affinities of variants over a large range of affinities. And moreover, all of these studies, the DMS, the deep mutational scanning experimental data, provide us only with a partial coverage. Here I'm showing an example from a paper from a few years ago where they tested the coverage of 31 DMS experimental data sets, and they showed that in only a few of these data sets we get a complete coverage, but in most of the data sets data sets, we get a partial coverage. So machine learning and algorithmic and computational methods can help us fill in the gap of this incomplete coverage. We used in our study a specific case study, which is the NTIMP2 inhibitor and the MMP9 protein, this complex. It is a high affinity complex, so it will help us in showing and demonstrating that we can predict over a large range of affinities. NTIMP2 is the inhibitory domain, and its interface is highly tolerant to mutagenesis, so it makes it an ideal candidate to test our approach through the deep mutational scanning data and followed by machine learning models. Here's the library and illustration of the library that was used in the experiment. I'm showing the NTIMP2 library. So you see the NTIMP2 here with 7 positions highlighted. These are the positions that were mutated in the experiment. They are crucial for binding, they're highly tolerant to mutagenesis, and they provide a broad range of affinities. So this is one library with mutations in NTIMP2 of these 7 positions. And a unique approach that my collaborator used was to insert an alanine at the N-terminus of NTIMP2 so that this insertion reduces the affinity and enables the measurement of low affinity interactions, which are usually missed when you do this type of experiments because you mostly measure high affinity experiments. So now we have 2 different libraries in the experiment, both an NTIMP2 library and what we call an ALA-NTIMP, where we have an insertion alanine at the N-terminus. So the goal in this study was to develop an approach to map the interactions and identify both unobserved affinity enhancing and affinity reducing mutation. And the approach that we're going to take is to train machine learning models, specifically deep learning, on the data that is going to come from the DMS experiment. And we were inspired by the study from the lab of Anthony Gitter that was published a few years ago in PNAS. So here are the 2 libraries that I just referred to, the NTIMP2 with many mutations, ALA-NTIMP2 with the alanine insertion. These were put through FACS sorting. This is one experimental technique to perform the selection for a specific function, where my collaborator both took care of the expression, making sure that the variants were expressed. And then you see gate 1 are the high affinity variants that were pulled. Gate 2 are the medium affinity ones that were selected, and gate 3 gives us the low affinity variants. And each one of these gates is then sent to high throughput sequencing. And eventually we can compare the frequency in the different gates, and then we can get a label for high affinity variants and low affinity variants. The labels that we used are defined in the following way. First, we calculated the frequency of each variant. Mut I is a variant number I. We counted how many reads support that variant, and we divided by the total number of reads, so we get the frequency. Then we defined the normalized frequency, where we divide the frequency of the variant by the frequency of the reference, of the wild type. So everything that we have in the labels is done in relation to the wild type. And eventually we defined 2 labels. One was the enrichment ratio that refers to high affinity, that we take the ratio of the variant in gate 1 over the normalized frequency of the same variants in gate 2. And for the low affinity, we took the ratio between gate 2 and gate 3. These are the 2 labels that we got from the experiment. And now we're going to train a model to predict these 2 labels. So how does the data look like for a computational person like me? I go back to the amino acid sequences, and now I highlighted in red the amino acid position that were changed and mutated in the experiment. So of course, the machine learning doesn't need to see the constant positions. So we remain with only 7 positions where you see in the data sets. And also each variant may have at most 2 labels that they just defined, but in some cases we may not have observed that variant in gate 1, 2, or 3. So for example, you see the NA where we don't have that variant. And here are the numbers of the variants that were observed in the different experiments. So we divided them by the number of mutations in each variant. So for single mutations, just one amino acid mutation, you see that the 98 with ALA library and no ALA is 84. Well, the number of possible mutations is 7 times 19, 133. So you see that even for single mutations, we don't cover all of them or observed all of these single amino acid mutations in the experiment. And of course, the number of possible mutations grow exponentially with the number of mutations, so we won't be able to cover all of them, but we do get some coverage of double mutations, triple mutations, which were errors in the synthesis, but we got it for free in the experiment. So in total, we got one data set of 466 data points, another data set with almost 2,000 data points, and these are the data sets that we're going to train the model on. We performed the partition of the data, 80% train, 10% validation, another 10% for testing, where we made sure that the validation and test variants had high coverage, high read count, so we can count to their labels. When in the training, they had the low read coverage, all the remaining variants. Here is the architecture that we used. We took the 7th position, we performed one-hot encoding. There you see a multi-layered perceptron with two fully connected layers. We also tried other approaches, RNNs, CNNs, protein language models, and none of them gave better performance than this simple model, so we stayed with it. If you are interested, you can later look at the hyperparameters that we searched. Eventually, we tried different 100 random combinations of 100 parameters. We chose the best one on the validation. We also used the random ensemble initialization technique where we train 10 different models, different initialization of the weights, and we take the average of the models in prediction time. So now moving to the results. First, we tested the model in cross-validation, meaning we held out 10% of the variants and tested how well we do on those variants. So first, I will show you how it performs on the NTIMP library. And here the test set includes 188 data points. It performs very well with a correlation of 4.96. So this seems like an easy problem, at least learning from enrichment ratios and predicting on enrichment ratios in the same experiment. But now let's move to the NTIMP library, where alanine was inserted, where we have fewer data points, only 466. The performance is not as good. The correlation drops to 0.82, but still I think it's reasonable, and we have some prediction power over this library, which is probably more noisy. But now for the more challenging task. And this is to predict on an independent data set. And this independent data set was composed of 26 variants, which we held out. And my collaborator performed experiments where he measured the absolute affinities of those variants, which are represented as Ki values, this association constants. These Ki values are really difficult in terms of experiments to get, so that's why we have only 26. Let's see how well the enrichment ratio predictions from the model do in predicting the Ki values. So we start with the NTIP model. We get a correlation of minus 0.5, and the fact that it's a negative correlation is fine, because Ki constants, when they go down, it means that we have increased affinity. Luckily, we are on the lower side of the 0.05 threshold, so we got a significant correlation. So we have some predictive power, but it's not as high as we saw in cross-validation. One reason that can explain this is because the variants are in different environments. In the high-throughput experiments, the DMS experiment, they were in yeast surface display. Here, they are in a much more pure environment where we measure the absolute affinities. Now we use the NTIMP model, and this model couldn't really predict significantly the Ki values. And another reason to explain why it doesn't do as well, which we also saw in the cross-validation, is the fact that the alanine insertion does not apply to the variants that we tested the Ki values for. So it's not the same variant. But when we mix the two models, we get an improved prediction performance with a correlation of minus 0.55, where the mix is just the summation of the models. Now, we wanted to do a more complex mix of the models with parameters, but unfortunately, we have only 26 variants. So we couldn't do something that was parameterized or more complex than just a simple summation. Now moving to some interpretation and knowledge of biology, we performed a mutation map analysis, and this is a very established and common technique where we predict the effect of each single amino acid mutation. Here you see the different 7 positions and all amino acid. Now, if we use the observed data, we wouldn't get the full map, but now using the model, we can get the full map. And what we see here are that we can identify 2 cold spots. These are positions where most of the mutations would increase the affinity. And we also identify a hotspot. This is a position where we see that most mutation reduce the affinity. And this knowledge can help us now design new inhibitors with improved function if we want to use them for therapeutic applications. The last analysis we performed is asking what are the characteristics that make deep mutational scanning data good for machine learning. So we tested two characteristics. One is the size of the data set, and I'll show you how we modify the sequencing depth. And the other is the variability in the DMS dataset, where we defined variability as the number of mutations in the variants. So to see the effect of the number of mutations, we held out a test set, high quality test set from the data. Then we sampled 350 variants with different number of mutations. You see in the x-axis only one mutation, 1 to 2 mutations, and so on. So we limited the number of mutations we can see in each variant. And then we perform this 10 times so we can compute an average with some standard deviation. And here are the results. So as you can see, training the model on just single amino acid mutations really deteriorates performance to below 0.6 in Pearson correlation. When we increase the model so it can see in the training data both single mutations and double mutations, we get improved performance. And only when we increase it to 3 mutations, we see it goes closer to 0.9. Meaning there is some interaction taking place between the different positions, some epistasis effect that we need to provide in the training data so the model can perform and predict well on unobserved variants. So this is something important that we should keep in mind when we design the libraries for the experiment. Another thing that we did was to see the effect of the sequencing depth of the dataset size. Here what we did, we just went to the FASTA-Q files and we randomly sampled 10%, 20%, up to 100%. So this is an in silico simulation of reducing the sequencing depth. And we repeated this 10 times. Here in the x-axis, you see the percentage of reads that we took from the FASTA-Q files, and also the number on average of the variants that we observed following this reduction. So for example, at 10%, you see that we got an average around 320 variants. And here, for example, you see that we don't see a significant reduction in prediction performance. Even with 10% of the reads, we can get to a Pearson correlation that is 0.9. And I speculate the reason for that is that while we increase the sampling noise of the different variants and the information that we measure, once we insert them into the machine learning and train a model on them, we average the sampling noise out. So we don't get such a significant reduction in performance. And now I can go to my collaborator and tell him, you can save 90% of the money on the sequencing. Because you can just do 10% of the sequencing or use the sequencing to give us more variants. As you have shown in the previous slides that I want to see more variants in my model. So future plans of this work, we want to improve the model by adding additional features. As I've shown you, we only use sequence features. So we want to include structural-based features. Here I'm showing an example of a contact map or structured graph, so you can provide a machine learning model. Evolutionary conservation is another type of features we can use. We hope to improve prediction performance by having more KI values, but we are limited if an experimentalist to do these experiments. Hopefully, if we have enough KI values, we can have a mixture of the model that is parameterized, maybe do transfer learning. But with the number of that we have now, we cannot do it. We want to also analyze the data in a way that we look at the interactions between the positions and perform an analysis on multiple mutations. Want to collect more DMS data sets and to develop a method that will be applicable to many data sets, and hopefully succeed in the application of utilization of protein language models, because they are very successful in other scenarios, but we weren't able to use them here. So conclusion. We showed that we can predict the affinity of unobserved variants, spanning a broad range of affinities. It can help other PPI protein-protein interactions and also help with therapeutic applications. We can apply this approach to many DMS datasets, and we also improved our knowledge of the NTIM2 protein inhibitor and its interaction with MMP9. I want to thank you for the attention, the funding agencies, and the student here is highlighted in the red rectangle. So thanks.
Thank you. Thank you very much, Yaron. We have time for one question. Just before that, I'd like to remind everybody here who's interested in CAFA4 results that we finally have them. There's a poster in B36. Those of you participating in CAFA4 will receive an email requesting co-authorship as usual in the CAFA community. It's nice that you segued into large language models, because after lunch, this is a lot of what we're going to talk about. And finally, I don't see Gonzalo here, but I think that frustration models that he talked about earlier might be relevant to what you're doing in terms of protein-protein interaction predictions. So we'll try to see, hook you up with him later. And Rafi, we have only time for one question, so Rafi, please.
Yeah, I was going to mention that the dynamical signatures that we calculated, we used to predict the MS functional data may be useful for you. But the one point that I wanted to make to you is that one criticism that we had when we were publishing that by some reviewer, I don't know who, was that we had a bias when we were testing by testing on data that was trained on, not because it's the same variant, this is an obvious thing to not have, but by having the same position in the training and the testing. You alluded to it somehow at the end, but simply withholding 10%, but it may be in the same position, there is a strong bias that is inflating your accuracy in testing of the method. So this is a criticism that you might have if it falls in the same reviewer that we had.
Yeah. So yeah, as I said, the partition was based on high-read coverage, but not a partition that partitions positions.
That makes the testing much easier if you train on the position and test on the same position, even if it's a different variant.
Yeah. Thanks.